Namaskar students, welcome you to our channel Bio by Gulab Sir. As a part of series of lectures, today's topic is enzymes. As you are well aware about enzymes as a biocatalyst, very often you hear about activities of enzymes and we use this word regularly in day-to-day life, enzymes. Now let us know about exactly what are enzymes. As in protein, we learn tertiary proteins are mostly enzymes. The tertiary protein, what they have, 3D structure, enabling them to be a catalytical molecule, and hence all the enzymes are protein, or those protein which catalyze biochemical reactions are called enzymes, except only one, that is ribozyme for NEET. Ribozyme is not a proteinaceous enzyme, and therefore it is something anomalous or different, and that's what you have to remember. And therefore in my notes I have clearly mentioned that all the enzymes are protein, but ribozyme is not a protein. Therefore it can be the point of attraction to us in NEET. Therefore please remember. So enzymes, they are catalytical molecule. They catalyze the biochemical processes. They have 3D structure. In that 3D structure they have substrate binding site. This is the active site where substrates are recognized by that enzyme. Enzymes are specific in their action. They catalyze only specific process. They won't catalyze other than that process. And therefore it is said that enzymes are specific in their action. Suppose pepsin as an enzyme catalyze the digestion of protein into peptones. Do not catalyze the process of digestion of starch or lipid. And therefore they are specific in their action. For example, salivary amylase is an enzyme to catalyze the process of breakdown of starch into glucose or maltose, but not to catalyze the process of breakdown of protein. And hence that enzymes, they have their specific substrate to act with them to produce the specific product, and hence we say that they are specific in their action. Enzymes never been get out of their specificity, and therefore that reactions which occur, they are very specific with their specific location for specific functions. So here what we have to learn with structure, that they got 3D structure. They have substrate binding site. They have specific cleavages with specific shape and size to accommodate specific substrate, and therefore only and only those substrate which supposed to be associated with that enzyme get into that cleavages to have that bonding between that substrate to form the specific product, and hence lock and key mechanism came into a highlight to explain about the function of the enzymes. Now before starting what is lock and key mechanism, we should understand what are the properties of this enzyme and what exactly enzymes about which we have to learn. I made this note for you. All the enzymes are protein except ribozyme. They are also known as biocatalysts because they only catalyze biological processes, not other than biological processes. As you know that there are also chemical catalysts like MnO2, but they are chemical catalysts. They do not catalyze biochemical process, and enzymes do not catalyze non-biological process. Enzymes are produced within the living organism and catalyze only biological processes. They are specific in their action, that I did explain to you. They cause to have the specific product. They are active at specific temperature. They are mostly active at 37 degrees Celsius. Almost all 99% of enzymes, they are active at 37 degrees Celsius. Therefore, all the living organisms, cold-blooded or non-cold-blooded, cold-blooded and warm-blooded, both they tend to have their body temperature at 37 degrees Celsius. You know that cold-blooded animal, with the change in environmental temperature, their body temperature changes, and therefore to keep 37 degrees Celsius temperature, they either hibernate or estivate. That you are aware. Therefore enzymes are active at specific temperature is very important. There are exceptions. Some of the thermoacidophils, that bacteria like Thermus aquaticus is an bacteria which has enzyme Taq polymerase active at 90 degrees Celsius. Can't imagine that some of the organisms, how they are evolved with some thermophilic enzymes, that they are active even at higher temperature. Otherwise, we know that protein get denatured at higher temperature. That means enzymes are protein and they also get denatured at higher temperature. But still, some of the organisms exist in the world that they have developed the capacity with their biomolecules to have the action of this enzyme even above 37 degrees Celsius, that too very above 90 degrees Celsius. And therefore, Taq polymerase is being utilized in polymerase chain reaction to have the multiple copies of enzymes. That is the only enzymes can work in polymerase chain reaction to have the billions copies of genes. It is miraculous. So some of the exceptions are there and they are miraculous. So otherwise enzymes are active at specific temperature. Now another point. Enzymes also active at specific pH. They are bound to be active at that specific pH, with exception that pepsin is active with 2 to 3 pH in our stomach. Because of that acidic environment, that pepsin has to be active at 2 to 3 pH to break protein into peptones and peptides. Otherwise, rest of the enzymes in our body, they are active at alkaline pH, that is more than 7 pH, 7.4. So 99.99% enzymes in our body, they are active in alkaline medium except pepsin, which active at 2 to 3 pH. That means they are active at specific pH. That is also very important. Pepsin do not act in alkaline medium and other enzymes do not act in acidic medium, and therefore specificity. Next point. Rate of enzyme double or decrease after every 10 degrees Celsius in either increase or decrease. That means with the increase in 10 degrees Celsius, that rate of enzyme becomes double, or decrease in 10 degrees Celsius, rate of enzyme become to the half. And therefore I made and said this, the rate of enzyme double or decrease by half for every 10 degrees Celsius either of side. That is means increase or decrease. I hope you got it. Now come to the point. After that, specificity or some of the specific character of enzymes. What is lock and key mechanism? It can be asked for 3 mark, or this can be asked in NEET. What is transition state? Or one of the following event in given diagram is a transition state, A, B, C, D. Can be in a choice, A and B, B and C, C and D. So B and C, they will be labeled A, B, C, D. What will be the answer? B and C are the event are in the transition state. Then what is the transition state? It is the state where neither substrate are free, neither nor product is free. Here in the first stage, substrate and enzymes are free. In the last stage, product and enzymes are free. But in middle, neither substrate nor product is free. Both are associated with enzyme. That is in the stage of process. So the process, ongoing process, is a transition state. Now what is happening with that stage? That is very important. Now once again, with first event, that substrate when come in contact with enzyme, they form enzyme substrate complex. Now to know about the speed of enzyme, what is the role of enzyme, that is very important. Let's see, it is like an agent. Suppose one has to sell something, the same thing other has to buy, but they are not in communication. एक को एक चीज बेचनी है और दूसरे को वही चीज खरीदनी है, लेकिन वो शहर के 2 साइड में रहते हैं, दोनों को कांटेक्ट नहीं है, तो बेचने वाले को खरीदने वाले के पास जाने के लिए कितना समय लगेगा, या कब तक वो दोनों को मालूम पड़ेगा एक बेचना चाहता है और एक खरीदना चाहता है। लेकिन वही अगर एजेंट एक शहर के बीच में बैठा हो, बेचने वालों को पता है कि वो एजेंट बेचता है और खरीदने वालों को पता है कि वो एजेंट के पास मिल सकता है, तो बेचने वाला एजेंट के पास जाएगा, खरीदने वाला भी एजेंट के पास जाएगा और इजीली खरीद और बिक्री हो सकती है। वैसे substrate A and B, they are present in cell far apart, being molecule, they are 100 km of distance. If we consider with their size, how long, then how much time they take to come together. Therefore, the rate of enzyme the rate of reaction without enzyme is very, very slow. That is 200 molecule per hour. But if same reaction is catalyzed by an enzyme, I am going to give example of carbonic anhydrase later, that 10 million time reaction become faster. That means 6 lakh molecules of that product produced per second. Such a alarming rate, miraculous rate of enzyme to carry out the process. And therefore, enzyme has very high role to increase or enhance the rate of reaction. by lowering the activation energy. We are going to learn. So enzyme substrate come in complex. So immediately the two substrate come in contact to form the bond between these two and how the product, and that is what lock and key mechanism. This is act as a lock, substrate as a key. When they come together, they form the block. That is what substrate enzyme complex. In process, bond between these two substrates is formed. Here it is clearly shown. Between A and B, the bond is formed. So with that process, after formation of bond, that means product is produced. Now enzyme is ready to release that product. After release of that product, as product AB from A plus B, enzyme become free for second round of action. Now here why it is called as lock and key? You just see that product as a key, and here it is lock, like a lock, and that structure appears so. Otherwise there is no any meaning or relation of lock and key with enzyme action. Simply that product seems to be like a key, and enzyme look like to be lock, and therefore it is called as lock and key mechanism. Otherwise there is no any other relation. I hope you got it. This diagram you have to practice so that you will come to know what is lock and key mechanism. I have given some steps of that lock and key mechanism. This you can use as notes, which same I spoke out. Therefore no need to repeat. So please go through it. You will get it. Now very important second part of this, or second part or paragraph of this explanation is, when I say that the rate of reaction is increases multifold, then how? That has to be understood. So with this graph we can understand. See, these are the substrate, and this is the product which produced from that substrate. On y-axis it is shown that potential energy which consumed for the reaction to produce the product and the speed of reaction. Here this substrate AB combined in absence of enzyme and produce the product, but the energy which required to produce that product is more than that of energy which required to produce that product in presence of enzyme, and therefore it is shown very clearly there. See here, this is what activation energy which required without energy to produce the product, and this is activation energy which required in presence of enzyme to produce that product. Then question comes, what is activation energy? So here the first point, that energy required to proceed the reaction is called activation energy. Now here it is clear, to proceed that reaction and produce product in absence of enzyme, activation energy which required is very high, and to produce that product in presence of enzyme, activation energy required is very low, and therefore the example that many reactions happen in our body are very fast. Therefore we have very fast response to the stimulus. If this reaction could have or would have not been faster, then we would have been very slow to respond to the stimulus. मैं पढ़ा रहा हूँ, आप समझ रहे कितने फास्ट प्रोसेस हो रही. अगर वही एंजाइम नहीं होते तो मुझे पढ़ाना और आपको सुनना बहुत स्लो मोशन में चलता. क्यों? तो टेक एग्जांपल how that reactions are slow. For that I am giving one example that is given in your book. See, suppose we inhale oxygen. No doubt during respiration carbon dioxide is produced, and that carbon dioxide is supposed to release first product which is supposed to form H2CO3, later that carried to the lungs where it split into H2 and CO2, then we release. That you learn in cardiac circulatory system. But here as a process, that carbon dioxide combined with water to form the carbonic acid in presence of carbonic anhydrase. See the rate of reaction in absence of, that is only 200 molecule per. So here it is shown that in absence of enzyme the reaction is very slow, which about 200 molecule per hour. But same in presence of enzyme, how many number of molecules? 6 lakh molecule per second. 200 molecule per hour and 6 lakh molecule per second. See the difference. That is what 10 million times faster than that of without enzyme. That means enzymes has very great role to how fast respond to the stimulus. If this could have not been, then we would have not been intelligence. We would have not been intelligent enough to how what we are today. So in the evolutionary sense, in the evolutionary process, why the organisms are becoming more and more smarter and more faster, more and more intelligent, that is what the role of enzyme, that helping that organism to have the fast reaction to respond to the stimulus, and that is smartness. It is because of enzymes. All biochemical reactions required for every response are catalyzed by enzymes. If that could have not been by enzymes, then that response would have been slow, and therefore I told you that enzymes play a very vital role to have the fast reaction, and that is very clearly mentioned here. You can see that carbon dioxide combined with water to form the carbonic acid. Without catalysis of enzyme, only 200 molecules per hour are produced, but with the enzyme, in presence of enzyme, same reaction becomes very faster and 6 lakh molecules per second are produced, and therefore you can imagine that for every response, some total of enzymatic reactions or some total of biochemical reactions which are required, if they would have been slow, what could have been happened? And because of this enzyme, we are very smart to respond to the stimulus faster as much as we can, and therefore we say that all species are evolving, and that is what the role of enzyme is very, very important and vital for all kind of species, all kind of organism to evolve with their intelligence. I hope you like this video. Also, I appeal you to share these videos with your friend, and if you are new, please subscribe it and press bell icon so as to receive new videos as and when they are uploaded. Thank you. Enjoy learning.